we take all together the HLA predictions or genetic predictions and then cytokine prediction and then some others on the T, can we make a score and try to put all that together and cluster the patients into a better score than what it is? What do you think? I mean, because you are all focusing on one element of the system. Or is it not a good idea?
I think eventually we want to do that. But still too early to say. Everybody focusing on yet another aspects of the immune system or another clustering of patients. You did cytokines and clustering, I did the T cells, and you do the genetics, but finally, of course, it comes all together. But of course, that needs a lot of time to work it all out and put it into one paradigm.
Yeah, but if we do these on the same large sample, so we can do what it's called system biology, and but not in different settings, because
you need to have the same cohort and then to work on that.
Yeah.
You insisted on the profiling of the immune system, I mean the phenotype of the immune system. But of course there is a repertoire, which is the other variable for both T cell, T subset, and B subset. So do you have any data or how to integrate those two aspects? You have the phenotype, but you also have for the T cell and the B cells, and also actually some of the neutrophil, the possibility to study the repertoire.
Yeah, in effect, I've not so been, let's say, what I studied in immunology has not been the repertoires, or I'm not a repertoire man. Of course, I know the importance of the whole thing, but I was more always studying immune regulatory aspects of cytokines and cells and how they interact. So of course there is that element in the immune system which needs to be seen. I have the feeling though that autoimmunity is absolutely normal. So the repertoire in the thymus, for instance, is made to recognize all autoantigens. And I think also with regard to what the Israeli group Michael Schwartz shows, actually, is that you have lots of MBP-specific T cells generated in the thymus, which actually are important for building up the brain. So these are the protective autoimmune cells. So there comes the repertoire in, I guess. Maybe some people are better in making good-fitting cells also towards and having this ability, while others are making the poor ones, maybe due to an MHC abnormality where you easily go wrong or not making the proper cells or not the most high affinity cells. But I have never studied it, but it's certainly open for study, and certainly when you look at the HLAs.
I agree. The repertoire also can be shaped by the genetics, the HLA and other genetics, but also environmental factor and things like that. So that could add to your very nice studies, you know, different parameters. And if we want to end up with index, I don't think it will be an index. It will be an algorithm probably, and I don't know if our intelligence will be enough. We may need artificial intelligence to get that. But thank you.
Talking about research and how to find the best biomarkers, but I think, I am not a physician, but I think we have to always think about how easy would be what we find in the laboratory could be used by physicians to address the questions they have in their clinical practice. And if I understand correctly, and I think all of us can imagine this, when a patient with a psychiatric condition go to see the physician has to make a decision quickly, right? And if it is possible, a cost-effective decision. So I think it's nice, of course, to explore all the ways we have been talking about, but I think at least in the next 5-10 years, maybe we will have also to think about how easy it would be to translate what we find in the clinics to help the physicians, the psychiatrists, to take the decision very quickly, because the patient cannot wait until they are treated. So I think this is something to remember.
That's why I was talking about score, because then you could think of something like that. Do you have any study related to family grouping? I mean, because you might have some similar disease in the same family, for example, one family.
As I told you, I have two cohorts, one with the children of a bipolar parent, which is in the family, of course, and the other one is twin studies. We're trying to figure out what is environmentally driven mainly and what is genetically driven. Of course, the various components of the immune system, it's quite different. For instance, Treg cells and Th17 cells are very much genetically driven, while the Th1 are environmentally driven. And the only association with monocyte inflammatory responsiveness, so with an inflammatory state, that is actually common environment in most of the twins. But I think the most illustrative twin is the monozygotic twin. The one is bipolar and has no thyroid disease. The other has thyroid disease and is not bipolar. So they have the two ends of the spectrum with the same genes. So it's a monozygotic gene. So it shows the effect of the environment and so on. So it really clusters in families as well.
Well, in a few weeks from now, we will have in this house a symposium on microbiota. So I've seen that some of you put the microbiota on the slides. I think it has to be put as the member of the game, certainly, because it's maybe one of the major players. So this is one more polymorphism which we should possibly study and combine with the others.
Actually, I have a question to Ryad. Regarding, you talk about gene HLA polymorphism, and of course there have been many studies on the microbiota. And, is there any association with the type of HLA molecule and the type of microbiota that you have, or it has been studied, or we don't have the data yet?
But not yet. Absolutely not yet. There is absolutely no study of the. Yeah, absolutely.
I would like to ask two questions. I'm Philippe Nuss from Paris. The first question is about the potential link between immunity and vascular and energetic metabolism in the brain, because there are many publications on to what extent mental disorders are disorders of metabolism, of energetic metabolism, and is there a link between vascular, energetic, and immunity? And the other point is about the classifications. I belong to a group that like to divide what is called big psychosis disorders into what is called biotypes. And NIMH has developed a group which is called BSNP. Have you tried to connect between your results and the results of BSNP biotypes? Thank you very much.
Yeah, there's strong correlation between oxidative stress, cellular oxidative stress, and immunity, inflammation, and consequently with what you are talking about.
And regarding the second question, I mean, yes, of course, I mean, this is really what we would like to do. So we would like to characterize our clusters better. First, the first episode psychosis cluster, but also the one that we found across categories. I hope that Stéphane Jamin would like to do this with us on the transcriptomic and the genomic data. And then, you know, any other biomarker that could be of interest, we would be very happy to look at the patients to see whether we can better characterize from a biological point of view these clusters. Of course, this includes also microbiota analysis, this includes neuroimaging studies, and so this is what we would like definitely to do in the next few years.